Okay, so in this video we're going to show how to prepare complete cell culture media that we use for Hackett cells. This media we use for many other cell lines is basically DMEM with 10% FBS or fetal bovine serum and Penn strep L-glutamine. So serum serves as a source of growth factors to allow cells to grow. Penicillin, streptomycin are antibiotics. L-glutamine is an additional amino acid that helps cells grow at a faster rate. So we add both of those things to the basal DMEM. And so the first thing to do is basically thaw everything out and warm it up. So I've already have it warmed up in the water bath here. We've got a 50 ml bottle of FBS, or aliquot of FBS. The Penn strep L-glutamine. We label PSG for Penn strep glutamine. Some of that will precipitate, you'll notice at the bottom of the tube, so just shake it up, it'll dissolve. And then we have a bottle of basal DMEM. All of these things are stored in various fridges in the lab. So in this fridge here, we have the FBS and Penn strep. So again, these are the aliquots of the Penn strep L-glutamine. These come as so we have any here. We have Penn strep L-glutamine, 100 ml bottle that we aliquot to 5 ml aliquot, since we need 5 ml per bottle of media we make up. We also have FBS, again 50 ml aliquots of FBS. These come in 500 ml bottles. And we use Cytiva fetal clone 3. So we basically thaw this out, aliquot to 50 ml tubes. So basically it's every single, to make up one bottle you need one aliquot of the Penn strep L-glutamine, one tube of the FBS. And then we have basal DMEM that's stored in this fridge over here. So we have, I'm not going to open the box, but DMEM high glucose. And high glucose is typically what we use. We have low glucose for other experiments we do, but high glucose cells grow at a little bit slightly faster rate. So again, the media is stored at 4 degrees. The other things are stored at -20. And so those things need to thaw out, and the media needs to warm up. And once we're done, we'll bring it into the cell culture room. Set this stuff aside for a second. And then open up the cell culture hood. Turn the light on. And then we want to do is spray everything down with ethanol. Again, anything going in the hood we want to make sure is sterile. And so again, spray everything down with ethanol before you bring it in the hood. If you have excess stuff dripping off, you can certainly wipe it up with a Kim wipe. And then what we want to do is open the media. There's usually a part here that will break down, so we rip that down. Trash. And we're going to want to add, basically just add everything to this media. So we're going to add 50 mils of FBS. This will put it in biohazardous waste. It's an animal product. The Penn strep L-glutamine, we'll add that as well. This could just go in the regular trash. And then what we want to do is label them that we've added things to the media. If you leave it unlabeled, how do you know it from a bottle basal DMEM? So I usually do plus for plus FBS plus Penn strep L-glutamine. I usually put my name and the date I made it. So this is the 16th. Okay, now we just mix it up, and now we're ready to go. Now we're ready to use the media for feeding cells or splitting cells.